Hello everyone. So last time we were looking at human development. We said that human development takes place after fertilization. We said we have a zygote that has been formed when an egg and a sperm come together. And then that embryo in the first stages of development, which is the first 8 weeks, is going to be called an embryo. And then after that, it's going to be called a fetus for the rest of the pregnancy until we have birth. So embryonic development, we said this is the first 8 weeks of the development of the baby, of the embryo. And so we start with the first phase. The first phase, the phase of embryonic development, is called cleavage and implantation. So cleavage and implantation, what we're going to see is we're going to have that once the zygote forms, it begins to divide. We have mitosis of diploid cells, right? Diploid meaning we had a sperm that was haploid, we had an egg that was haploid, and when they came together, they made a diploid cell. So we have begins to divide very, very rapidly. And more importantly, without cell growth. And remember that this is still happening within the fallopian tubes. In the fallopian tubes of the female, that's where egg meets sperm. So we have the zygote that forms in there. And then as it's traveling and making its way towards the uterus, it begins to divide very rapidly without cell growth. This kind of rapid cell growth, this rapid cell division without cell growth is called cleavage. Okay. And then what we have is we have sort of the first stages of the embryonic development are going to take place. And so here, as you can see, we have an egg and then fertilization. We have a zygote with both mom and dad's nucleus. And then we see that this starts to divide, right? 2 cell, 4 cell, 8 cells. Remember that if it stays within this envelope, we have one baby. If it were to divide in half like this, we would have identical twins. And then this one would start to do this. And then this side would start to do this. And so we would get 2 babies that are identical genetically. So as it's tumbling down the uterus, eventually we get to a 16-cell stage called a morula. And then once it enters in the uterus and it starts to implant, we get ourselves into a blastocyst. Okay, so let's have a look at the stages. So we said we have zygote, and then we're going to go morula, and then finally we have blastocyst. And so in the zygote, here we have, it's just 1 cell. The morula, this is a 16-cell stage. And then once we enter the uterus, what happens here is we have fluid from the uterus diffuses in. Pushing the cells to one side. And so that's what you're seeing here. That is then our blastocyst. Okay, so that's what you're seeing right there. This is the fluid that moved in and is now diffused into our embryo. Okay, then this is when the blastocyst then burrows into the endometrium. Remember, the endometrium is the very thick lining of the uterus. So if I were to sort of draw the uterus, the uterine wall, okay, so that's the uterine wall, and what I would see is I would see that little blastocyst would burrow in, and so now it would be in here, okay, and once it burrows in, this is what is called implantation. Now is when you're technically pregnant. And this little tiny embryo, what it does is it starts to grow little roots. It starts to grow those little roots that tap into the mother's blood system, right? And so those are called the chorionic villi. We talked about it when we said we were doing genetic screening. So it grows little projections called chorionic villi that tap into mom's bloodstream. So this little embryo is really a little parasite at this point. It's taking nutrients from mom. Okay. Now, as an aside, what I like to do is I want us to have a look at that blastocyst. So what we are seeing, I'm actually going to use a picture, I think. Well, I think I have a yes, I do. So here is a blastocyst that you can see. Here we go. And what I want you to notice is the very outside cells are going to be called the trophoblast. And the cells on the inside, called the inner cell mass or the embryoblast, are also made up of the same genetic material. So let's say that we label these guys. So let's say that this is the trophoblast. And so those outside cells, they will become the membranes that support the fetus, right? So these guys will become the placenta, the amniotic sac, the umbilical cord, all of those things come out of the trophoblast. Whereas the inner cell mass, or that embryoblast, you probably guessed it just from the name, embryoblast, it becomes the embryo. Whatever organism we're dealing with. Both layers are important. They have the same genetic makeup. They all came from that zygote that divided many, many times. And so if I wanted to do some genetic testing, I could take a sample of these chorionic villi that come out of the trophoblast, and I would be able to do some genetic testing. So once these villi grow out of the trophoblast, if I want to test one of these cells, I can take the nucleus, check the genetic information to see if there are any abnormalities with my baby. I don't have to touch the embryo. I can leave the embryo alone. Okay. Now the next set of steps that takes place during this development is called gastrulation and tissue formation. So during the second week of development, then the cells of the embryoblast migrate to the middle of the blastocyst. So what I get is if I had the uterus, right, and I have the embryo implanted in there, it has grown those chorionic villi. Now what I get is I get the cells are no longer pushed to one side, but they're actually in the middle. So they look like something like that, okay? And then obviously there's other cells on the outside right here. And so what is starting to form is that amniotic sac, right? So this right here is that amniotic sac that is going to be protecting the embryo, okay? So we can make a little note there. So the amniotic cavity, oops, with amniotic fluid begins to form. Okay, so that's right there. That's the amniotic, or I should point it probably this way. Right? So right here, this space right there is the amniotic, not the cells in the middle. Okay? And then, while that is happening, the cells of the embryoblast that are now in the middle begin to migrate again and differentiate. So for example, my head cells move to the top. Okay, so that's what we get. And the cell are divided into 3 germ means to give rise, germ layers, okay? And so these 3 germ layers might not give me, nope, that's not what I want. Recent, this is the one that I want. For some reason, sometimes it doesn't show it to me. Okay, so this is what I want. And so the 3 germ layers are going to be the ectoderm, the endoderm, and the mesoderm. Okay, so you can see these 3 layers that are forming just in this middle portion right here. They're starting to divide, okay? And so what we get is we get the ectoderm will become the skin, and then the neural ectoderm will become the nervous system. The mesoderm, this will form your muscles, okay? It'll form your muscles, it'll form your skeleton, it'll form all your connective tissues, your blood, okay? Whereas the endoderm, this becomes the gut, so your stomach, your esophagus, your liver, and then your respiratory system as well. The mesoderm, one of the ones that is important to know is together with muscles, you're going to get the heart as well. So the heart too is part of that mesoderm, okay? So here, let's go ectoderm. This is on the outside. And this guy we said becomes skin and the nervous system. Then we have the mesoderm. This is the middle. And it becomes skeleton, muscles, and reproductive organs. And then we have the endoderm. And the endoderm becomes your respiratory, digestive, and endocrine system. I'm going to include a video for you guys to see how those occur, the development as well. Okay. And after we get to this point, what happens is the cell itself goes through a process is called invagination, and we get a neural tube so that this outside layer sort of pinches in like this. And so this becomes the skin, and in here you have the nervous system, the spinal cord and brain that starts to develop, and then eventually that closes like this. And so I have the skin on the outside, and I'm going to have the nervous system on the inside. That's how I get the ectoderm that is both the skin and the nervous system. Okay. After that, we are done with embryonic development. So next is going to come the fetal development. Now, while this is happening, these first few weeks, while this is happening, we're also going to have some growth, and also some, structures are going to start to develop. Morphogenesis just means we get the little hands and the face and the little feet. Okay, so then the rest is going to be the development of the embryo. So then one little aside again that I want to have is I want to talk about the extra embryonic membranes. All right. And the reason why I want to do that is because we looked at this blastocyst, and so we're looking at what the fate of this is going to be. But now I want to look at what the fate of this is. What happens to those outside cells? And that's what those extra embryonic membranes are. So the very first one is the chorion. I'm going to give them to you in the order in which they develop. So the chorion is the first. And the chorion, with its chorionic villi, it absorbs nutrients from mom right away. But the chorion also produces a hormone called HCG, human chorionic gonadotropin. And HCG is what is detected by your pregnancy test. And so I want you to think about that you would not have HCG circulating in your system whatsoever unless you had an embryo that had implanted itself in your uterus and it started to grow a chorion. Unless we're getting these, we're not going to have HCG. So you can't have a false positive. If your pregnancy test turns out positive, you have an embryo implanted in your uterus, okay? And what is the point of having HCG? It's not just for the pregnancy test to be positive, but also HCG keeps the corpus luteum around, and therefore maintains high levels of progesterone. And remember, we said that the way that you get your period is progesterone levels drop and you shed your endometrium. In this case, what we're having is the endometrium stays put because the levels of progesterone are kept high, okay? And this hormone is going to be secreted for the first few weeks. Until the placenta forms. And the reason for that is then the placenta secretes its own estrogen and progesterone, and I don't need HCG anymore to maintain the endometrium. The endometrium can the corpus luteum can go away and the endometrium will stay because the placenta will secrete its own progesterone to keep it that way. Okay. Now, the second structure that forms, I think I was using green, yes, is the amnion. So the amnion gives rise to the amniotic sac. And so this membrane filled with fluid called amniotic fluid protects the embryo from trauma and temperature fluctuations. When people say that their water breaks, it is the amniotic sac that breaks. And releases that amniotic fluid. Okay, so that's what that amniotic sac does. And then next we have the placenta. The placenta forms from the chorion. So it comes from the chorion. It just keeps growing. And this particular membrane is the site for metabolic exchange. Between mom and baby. Metabolic exchange, I mean carbon dioxide is exchanged for oxygen, nutrients are exchanged for waste. Okay? And we know that it produces its own hormones to maintain the endometrium. Well, the placenta is not going to do anything unless we have an umbilical cord. The umbilical cord is formed from a membrane called the allantois. Some people say allantois. I like the French. Sounds a little bit better. And so this, again, is the connection between the fetus and the placenta. And this guy contains veins and arteries for blood exchange. Okay, it can grow up to 60 centimeters in length, so it can be quite long. And then the last membrane that we have is the yolk sac. And the yolk sac, unlike in chickens, the yolk sac has no nutritive function for humans. This is a sac that contains early red blood cells because the fetus doesn't have a bone marrow. So yes, I need to transport nutrients from mom to baby and then waste from baby to mom, but I need blood to do that. That's how it gets transported. And so where do I get red blood cells if I don't have a bone marrow to make them? The yolk sac will give me that first little bit of red blood cells. And then the yolk sac starts quite large. And then as the bone marrow starts to form in the fetus, then the yolk sac gets shrunken. It shrinks more and more and more. Okay, so for that, now I'm going to show you. I'm going to maybe delete that and put it in the next page. Here we go. All right, so here you can see the different membranes. So I have the placenta. Okay, so you can see the placenta. The chorion is that first layer that forms everywhere, and then it eventually grows into the placenta. And this side, it gets quite large. And then the amnion with the amniotic sac can be seen right here. Here's the umbilical cord at the beginning, and then again, and you can see it has 2 veins and 1 artery. Okay, and the artery is taken away. Remember, arteries go away from right? A for away. So the artery is taking blood away from the fetus, so it's going to have the wastes and everything, and the veins are returning it to the fetus, so it's going to have the oxygen and the nutrients going to it. And then here you can see that yolk sac that eventually goes away once we have all the bone marrow that we need. All right, so these are the notes for the embryonic development. Next time, what we're going to be doing is looking at fetal development and birth, and we're going to be done with reproduction. So I hope this helped. And again, I'm going to put a video for the endoderm, ectoderm, and mesoderm for you guys to watch. So if you can, watch that as well. All right, we will catch you guys tomorrow. Bye-bye.